Bags. It is a great honor to welcome Professor Bruce Gale, Chair and Professor in the Department of Mechanical Engineering at the University of Utah, USA, and an accomplished entrepreneur. Professor Gale has significantly contributed in microfluidics, biomems, and biotechnologies, focusing on developing advanced systems that addresses critical healthcare and challenges. In today's session, he will be speaking on microfluidics, past, present, and future, offering us a comprehensive perspective of the evolution, current advancements, and the future potential of the transformative field. We are delighted to have with us today. Join me in extending a warm welcome to Professor Bruce Gale.
All right, we'll try this one. Thank you so much for the warm welcome. It's great to be here at REC again, and I look forward to interacting with many of you. Thank you for the invitation today, and I hope to give you some understanding about microfluidics that maybe you didn't have before. So I'm going to talk a little bit about, as it said, the past, present, and the future, but also about how we can use microfluidics to achieve scalable and affordable innovation, hopefully. So as we look at this today, I want you to think about how you might fit into this microfluidic universe. So I'm going to talk a little bit about how different types of engineering, different types of computer science might be of use. And as you can see in this image here, this is an AI image. I said, hey, AI, make me a picture of a microfluidic chip, right? And so this is not a real one, but it's kind of an idea of the different sorts of things that you might see in these devices. So I'll show you some real ones in a couple of minutes, but this is kind of a fun idea to think about the electronics, the optics, and other things that might go into a chip like this. So as I mentioned, I wanted to talk a little bit about how you might get involved. So this image that I'm showing here on the screen, this is a 3D printed device from a group that we work with at BYU in the United States. And they've 3D printed this device that can generate a pump and move fluids around on these devices. And as you can see, these are quite small. In this case, they're only 2 pixels wide. So 3D printers are something that I suspect may be a little bit more easy for you to have available to you than maybe some of the other technologies that are used for these sorts of challenges. So 3D printing is something that's being developed by a variety of different disciplines, and in microfluidics we need all of these different types of people to be able to develop our microfluidics. If you look at the automation of design of microfluidic systems, this is where software comes in. So I suspect many of you are involved in the software area, and a lot of microfluidics in the future is going to be developed or going to be determined by software. And so I am looking for software or people that are excited about software applications in microfluidics so that we can work together to solve some of these problems. But this is just an example of some software that we've been using to generate microfluidics. All right, so I'll take a minute now and just give you a background on what is microfluidics and where does it come from. So microfluidic devices, they tend to be, as you imagine, small. So we have channels normally less than about 1 millimeter. But most of the time these channels are about the size of your hair. So that's about 100 microns or 100 micrometers, and you can use these devices to move fluids around in a variety of different ways. These images on the left, we show different valves. We have one here where there's a sperm trapped in the middle. Anyways, you can develop a variety of mixers and other types of things that would allow you to use these microfluidics in a variety of different ways. So you can have a large number of applications. But the main application that we're looking for is how can we take these devices and learn more about you or about me, right? What we want to know what's going on in our body at any given time. So this is the personalized medicine. So you could take a blood sample and quickly determine whether you have COVID or cancer or diabetes or what your situation is, as opposed to maybe needing a large laboratory experiment to figure that out. So these are the sorts of things that we're working on, is how to turn your data in your body into information that a doctor or yourself can use to take care of yourself. So you may be familiar with MEMS, which is microelectromechanical systems. MEMS is a kind of a broad area, and we like to think that microfluidics is part of MEMS. And if you look at this diagram here, I'm sure you can't read everything there, but this just shows that there's sensors and other types of components that can be used in almost any part of the body to make a measurement and to understand what's going on, including some of these related to biology and the fluids in the blood, for example. So this is well developed, and the physical ones are growing to be found almost everywhere. For example, in your phone, you're going to have dozens of these sensors that are MEMS-type sensors. But I don't think anyone has a microfluidic sensor or a microfluidic device in your phone yet, so that's what we're working on, is how we can get one of those into something that everyone's using. So just very briefly, microfluidics has a long history, and we're not going to be able to go through all of this today, but you can see that most of this is developed most rapidly in the last, I'm going to say, 10 to 20 years. So that's this section here in the top half, where we have the development of organ on a chip, inertial microfluidics, and other arrays that can let us to develop information very quickly. So what types of applications are most useful in microfluidics? Well, these are going to be high-value applications. So that's something that someone would pay a lot of money for to understand. Right now, that includes things like DNA information about yourself. These are things like organ on a chip, where we can replace an animal with a microfluidic chip to help us understand what's going on inside the body. We can also use microfluidics to solve problems for individuals. So you don't have to be an expert anymore to do these tests. Now it's something I can just put in a blood sample or a sputum sample and run that analysis, and I get the answers very quickly. So you don't need a doctor or a lab analyst to do that for you anymore. Now, I want to point out that microfluidics has not been adopted as quickly as people expected. So when I started in microfluidics 30 years ago, everyone thought that these solutions would come rapidly, but they've been a little bit slow, and I'm going to talk a little bit about why that is. But the main challenge is every different device. So if you're trying to do a test for, say, diabetes and a test for COVID, these have very different processing steps that you have to go through to make them work. So that leads to, we have a variety of different microfluidic devices that have to be built, and they don't have a standard approach. So we'll talk a little bit about maybe how we can adapt to that as well as we use software. So very quickly, I'm going to talk about what are some of the different types of microfluidics. We have continuous flow microfluidics, where you have a flow that may come in at one of these inlets, and it's just going to follow this curve and exit at some point. So you just have flow going all the time. And the same with this device. It is going to generate mixed colors of these dyes that are shown in this particular device. But you can process larger volumes. This could be milliliters even of fluids, and you may say that's not that big, but in microfluidics that's a large volume. You can do things that are very fast and take advantage of what are called inertial flows, or you can do things very slow, and that is referred to as creeping flows. And then finally, you can provide the pumping on or off the chip. So these are the different types of ways that you can develop it. So if comparing continuous flow to what we'd call as discrete microfluidics, these are where you're going to have a sample where the flow is moving back and forth just by, in this case, pneumatic pressure. We're going to move the fluid to different locations, but it's a fixed volume. These are valves that are inside of this device, and they allow us to mix and match different fluids rapidly and inexpensively. So these are very nice because they can do sequential things. So if you have a test where, okay, I mix this fluid with that fluid, and then mix this fluid with another reagent, these are easy to set up, and can be used with a variety of different tests all at the same time. So that's what I'm going to call discrete microfluidics. The third area of microfluidics that's being developed is often referred to as droplet microfluidics. So as you can see here, we have droplets that are being generated, and in this case we're putting small beads into some of the droplets. You may be able to see them here, but as this shows up on the top, you can make different sizes and different numbers of droplets very quickly. Now, why would you want droplets? What's the value of droplets? Well, the main value of droplets is these are now reaction chambers. They're self-contained. You can put DNA or cells or whatever reaction you want, and it's trapped in this very small volume. And you can make thousands, millions of them very rapidly. And so droplet microfluidics is being very common now in DNA genotyping, digital PCR is the primary example, and DNA sequencing. So these are, it's becoming very common in those areas. So that's the third area. And one other example of these droplet microfluidics is you can put single cells in. So now I can analyze single cells one at a time as they come through this, this channel. So they move through, they pinch off into single droplets, and now we have, oftentimes you can have one cell per droplet and do an analysis on just that cell, and you can do PCR, amplify it, or you can look at whole genome analysis, transcriptomes. Anyways, there's a wide variety of different applications that you can use now with these droplets. Anyways, that's the third area, is droplet microfluidics. And then the fourth area that I'll talk about briefly is digital microfluidics. These are droplets. Instead of them being kind of free-floating in the solution, they are now moving along these paths, and you can choose which path you want them to take. You can mix and match, run these droplets into each other, and they can combine. You can separate them. Anyways, it's like a programmed microfluidic droplet, so you can move it around on a surface. Anyway, so those are the four general areas of microfluidics, but the question is, what are we trying to do with them? Well, as I mentioned, one of the core concepts is a lab on a chip. We'd like to take this microfluidic device and put in all of the different features that you might have in a different hospital lab. So that's kind of the idea that's being shown here. You take those features, put them onto your chip, and now we don't need that hospital lab anymore. We can take the hospital lab with us because it's now portable. And if you look at this one, again, this is that AI chip, just kind of explaining that idea that you can take that hospital lab and put that into that microfluidic device, so in kind of a fun way. All right, to make a lab on a chip, you're going to need a variety of different capabilities. So the very first thing that you have to be able to do is to collect a sample. So in most cases, this is going to be a biological sample. This may be a blood sample, urine, mucus from the nose, or sweat. I mean, there can be all kinds of different samples. So you collect the sample. We need to take that sample and prepare it somehow. That oftentimes means we're going to either extract DNA, maybe we're going to take out RNA, some sort of protein analysis, maybe we want to collect cells from it. These can be all the different reactions, and this oftentimes is the hardest part in microfluidics. We have to design a different setup for every different type of sample that we're working with. Once we have the sample prepared, we have to do some sort of reaction. That is kind of our general analysis, and then we collect that data, do some sort of measurement, and then that requires some sort of data processing. As many of you probably know, no matter what you're building at this point, you probably need software to run it when you're done. So I found that the software capabilities that we can develop really determine a lot of what we can do with our microfluidics. So briefly, I'm going to give you an example of a commercial device that's pretty close to this lab on a chip. So you kind of have a sense of what's coming from that. This is a company that based in Utah. It's called BioFire, and they make a film array. And there's an instrument, and then it has, it combines the sample preparation, the amplification, and the detection all in one. So it takes these three instruments and turns it into one. So that's kind of the lab on a chip, if you will. Now we've got multiple lab components. And this is the cartridge that is inside of it, and it does all of these steps, and I'll dive into that a little bit more. But it has a reagent storage pack, and then this, we're going to call it a chemical circuit board, which is made by laminating two sheets together, two pieces of plastic. So here's a close-up of this cartridge. This is where the sample preparation is done, and then the analysis is read out optically in these different wells. It reads 96 different things at a time. So it has this pouch, it goes into an instrument, all the molecular steps are inside of that, and then it should make this whole process very simple. So let's look at how that works. So inside this film array pouch, we have these kind of compression, or could be pneumatic, that pushes these chambers, the circles here with the fluids in them. It pushes down on them and moves the fluid between different locations. As it does so, it goes through 3 or 4 steps. It's lysed, these cells are lysed, the DNA is then pulled out, and then there's a nested multiplex PCR. So PCR stands for polymerase chain reaction, and that's used to amplify the DNA. And there's actually 2 different sets of PCR that's done in this device. This first well here does PCR 1, it's then diluted, and then they do a PCR again. And in this one, it's going to be looking for specific disease states, and I'll show you what some of those are in just a moment. So the first one amplifies just the targets in general, and then the second one looks for very specific sets of DNA. And then they use fluorescence-based melt curves to identify the pathogens. So that's, if you're familiar with that may be helpful. But so how do they use this then? So right now they have 4 different panels. So in one device, you show up at the doctor, maybe you're coughing, sneezing, or whatever the case may be, and they can take a sample from the nose or the mouth, and they put this into the device, and they can detect 20 different respiratory viruses or bacteria. So in this case, that would be some of the viruses that are listed here, including like COVID, SARS, RSV, etc. And now, instead of a doctor having to guess what you have, they can figure out right away, oh, you have, this specific virus, we know how to treat that, and it brings a lot of comfort both to the doctor and to the patient to know right away. And this takes about 45 minutes for this test. So this is one of their most common tests, is a respiratory panel. They also have one that's a blood culture identification panel, and what that means is they take a blood sample and then they look for infections or other things that you might have in the bloodstream. And then for the third panel they have is a GI panel. This is looking for infections or other viruses that you may have in your digestive system. So if you come in, say, vomiting or with diarrhea, you can quickly figure out which of these mechanisms may be causing that problem. And you can imagine that that's very helpful. So now we're looking for 22 targets all at the same time. There's also one for cerebral spinal fluid that can identify pathogens basically in your brain fluid. So these are just an example of one device that can suddenly look for 4 different types of diseases, just depending on which of the film arrays you're putting in. And it's a pretty good example of a lab-on-a-chip device at the current time. So anyways, I'm going to talk to you about a couple of other applications. I'm going to start with just some of the things that we do in my lab to kind of give you a wide variety of what you can do with microfluidics, and then I'm going to dive into some specific ones for a few minutes. First of all, we've been working on some chips that are just, I'm going to call them complex. This one has more than 300 valves in it, and these valves can move fluids around, sort particles, objects in the fluids, and then deliver them to a 96-well plate. So that's what's happening with that device. And these can be used for DNA analysis and other things with large number of samples. So I'm just going to call that a complex microfluidic system and pathogen detection. We've built devices to do digital PCR. In this case, you would put in a fluid here and you split it into 1,000 different wells and can do PCR to analyze and count viruses or bacteria that are present in a sample. So that's another example of from a biotechnology standpoint. We also have continuous flow PCR devices that allow you to do PCR in less than 1 minute, so it's very fast. So these are nucleic acid analysis and sequencing devices. In my lab, we also do cell and nanoparticle separations. This allows you to take particles or a sample and split it into its component parts so that you can identify the pieces that are relevant. So, for example, we do sperm separations from cell samples. And this is just an example of what that might look like. You can see that certain particles are being focused to the center, and other particles, the smaller ones, are off to the side. And so we can separate them based on their location in this channel. We do what I just call cell and small animal processing. We do things with zebrafish, embryos, little worms, C. elegans worms. We've also done a variety of medical devices. So these are like drug delivery devices. These are some microneedles that we've made. This one's a drug delivery device that goes in the eye. And then this is to attach two arteries together. So if you were to cut an artery in your arm or leg and needed to rapidly put that back together, that's what these devices are for. This is also a drug delivery device for nerve regeneration and local drug delivery. We have devices that will make arrays, so you can do a lot of experiments all at the same time. So instead of doing one or two at a time, you can do hundreds at a time. And then finally, I mentioned we've been doing some work with droplet microfluidics like we already talked about. So those will give you a little bit of an idea of kind of general applications that we are working on. And so now I'm going to take a couple of minutes and talk about, what is the state of the art in microfluidics, and what would you maybe want to use microfluidics for? So first of all, everyone should recognize that microfluidic physics is relatively well understood. So people have been working on microfluidics for more than 30 years now, and the fluid physics hasn't changed really as a whole for maybe 100 or almost 200 years. So the physics is well understood. So what does that mean? It means that we can use the physics that we have to analyze and develop microfluidic devices. So what are some of the physics that are maybe interesting at this scale? So let's start with mixing. So if you were to combine in a microfluidic channel a whole bunch of different colors, you can see that they literally don't mix. They just come in right next to each other and stay together in their own separate lanes and lines. Now that can be really helpful if you're trying to keep things separated, but if you're trying to mix those fluids, it suddenly became very difficult. So this is an effect of the flow is all laminar. So those of you that have taken fluid mechanics will recognize that laminar flow means it's kind of in layers, and you can literally see the layers here, right? There's no mixing of those layers. They stay together. If you go down far enough, they'll start to mix eventually, but it's through diffusion. So we don't have no turbulent mixing. There's no chaotic activity really going on there. Okay, because of that, the flow becomes deterministic. And what that means is I know exactly where it's going to be. When I do it today, it's going to be exactly the same as it is tomorrow, as it is the following day. So it doesn't change. I know exactly how it's going to work. I'm assuming they have bugs like this in India somewhere too, but this is a picture from the US of some of these bugs that can basically walk on water, right? So this is caused by surface tension. So you look at this space around this bug's leg, this insect's leg, and you can see that there's a little depression there. And that's caused that the weight of the bug is being held up by the surface tension of the water. And so as you get to smaller and smaller scales, it becomes more and more important. So this is a feature that we can use, but it can also become a problem if we ignore it. The other thing that happens is that inertia can typically be ignored. So if you are a bacteria swimming in water and you stop swimming, how far do you coast? Basically not at all. You're just going to instantly stop. So if you're a boat out on the water and you stop, you're going to kind of glide for a little while. But bacteria, they just stop. So inertia and things at the small scale, or inertia just is ignored at the small scale typically. And then the last thing is fluids appear to become kind of thicker, kind of like honey, right? It doesn't drip and it's not thin. It essentially thickens up. So those are kind of the key things that change when you get down to the microscale. And we can take advantage of that and use them for us, or we may want to, in some cases you may need to recognize that these could be problems if you're trying to avoid that. Okay, so the end of the message here is that microfluidics is well understood from a physics standpoint, and it's well known how these manufacturing processes now work. So it's mostly a tool. So if you wanted to just be a microfluidics researcher, so I've been doing microfluidics for 30 years. When I started, you could say, I just want to do microfluidics. But if you said, I want to be a microfluidics researcher now, you probably wouldn't get very far. And what that means is you need to have some other application. So when I say that microfluidics is a tool, it means you can use it to study something else. So I'm going to show you some of the something else's that you can study with microfluidics and use it to develop a career. So it's just important to recognize that it's not, maybe not a goal in itself now. So there's a variety of analytical tools that are well developed. Electrophoresis, as I mentioned, PCR. There's all kinds of electrical characterization, fluorescence, cell separations. All of these different tools have been developed and can be used with these microfluidic components. So that they're well known. And this device here is a PCR device. It's cooler on the bottom and it's warmer on the top. And as the fluids move to the top, they literally melt, and the dyes that are inside them fall apart, which is why it gets darker as it goes up. So they can use this melting analysis to characterize DNA. So that's an example of one of these tools that is well understood. We do know how to make all of these kind of general, like I said, electrophoresis, PCR, that analysis is well understood, but sample preparation is not well understood, and that means we don't know how always to take a sample and turn it into something that we can do PCR on, or that we can do electrophoresis, or that we can do some sort of other analysis. So we have to design our own microfluidic sample preparation for every single device. So if I'm trying to detect COVID, I have to do a different sample preparation than if I'm trying to detect even E. coli or some other sample. So that's maybe one of the biggest challenges, or I consider the biggest challenge, is needing to adapt for every different type of sample. So that's one of the challenges that we're going to want to address as we go along. All right. So maybe another question that you might ask yourself: is microfluidics standardized like MEMS or integrated circuits? So if you make a computer chip, there's only one really way that you make computer chips. You can go to any foundry in the world and they'll basically make it the same way. But microfluidic devices, you don't have that same option. There's very few, there are foundries for microfluidics, but if you go from one to the other, they're going to be totally different. They're going to be making something that's in a different way. And so that's actually a big problem, and that's basically what I point out here, that there's not really great standards for microfluidics, and that's because there's so many different ways that you may have to solve a problem. Sometimes you're going to need to make something in glass. Sometimes you're going to make it in plastic. Sometimes it'll be silicon. Sometimes it may be a 3D printed device. It could just be completely different. So there's no real universal approach to microfluidics, and so we need to, I'm going to argue we need to solve that. So I'm going to talk to you today a little bit about maybe some ways we can solve this problem, and maybe you can help us solve that problem. And as mentioned, because the quantities in microfluidics are not often not in the millions, it can be expensive to manufacture these devices. So we'll talk to you about just a couple of ways that we can do manufacturing in maybe a less expensive way to make devices as well. And one of those examples is here. Can we make simple and inexpensive devices? In this case, we have a multi-layer device that are made out of just sheets of plastic, and we can just cut them out with a laser or with a knife plotter, and they're relatively easy to make, and then you can laminate them together. So this can be done in some sort of roll-to-roll manufacturing process, which could be relatively inexpensive. The other thing that this device has is this component right here, which you can see. I'll show you the next part of this slide. This component right here is designed to pump the fluid. So it's a wicking material. It's a capillary pump. So it's kind of like a napkin or a piece of paper. So as you put fluid in here, it's going to drag the fluid here and pull it into this chamber. And this chamber is full of this paper. And as it does so, it actually keeps a continuous flow here that doesn't change. This shape actually generates a continuous flow. And in this case, this is a cap. You put your sample in here. You put the cap on, and it pressurizes that fluid, which pushes it into this capillary bed, and now it pulls it across. So now I don't even need electronics to drive my device. And in this case, we have a silicon chip sitting on the bottom. So I'm going to go back to this. You can see that there's a space here that's left for the silicon chip to go in. And now I can have a smart chip that does some sort of optical or other measurement in a relatively simple device. So the microfluidics can be made very simple, and now the electronics can be a little more complicated as it needs to be. So as you can see, this device was made in plastic. It doesn't require any power, any electronics, and you can do a test for a variety of different biological phenomena. So, and these are some of the main methods that are used for microfluidics. So looking at some other options, what's going to drive microfluidics? Well, one of the main things is point of care. We would like to be able to do tests wherever we are, right? So if you're out in the wilderness, in the desert, out at sea, we should be able to make measurements relatively easy. So we're trying to build microfluidic devices that can be, say, attached to your phone, attached to a small instrument so that you can take it with you and do this analysis. So we need creative people like you to help us design these point of care systems that will allow us to make these tests wherever we may be in the world. It would be ideal. So right now with your phone, for example, you can measure maybe heart rate or some electrical sorts of signals, maybe even pressure on occasion, but there's no real fluid analysis that's done on your phone. So those are the sorts of things that would need to be developed. As we look at some of the commercialization challenges that come with microfluidics, some of the devices that are being made can be very complex. And this device right here it's made by Fluidigm. It's an integrated flow circuit, as it's called, and it has a lot of detail in this space, which it kind of shows up here if you kind of zoom into it. But all the samples are put in the outside. But this is an example of a complicated commercial microfluidic chip that's being sold today, but it can only do one type of thing, but it does it very well. So how do you get around that? Well, one of the ways to solve this problem is to do modular design. So you may have a board. In this case, it would be like a printed circuit board, and then you have different modules or chips that you can put onto this board. So if you're familiar with printed circuit boards, these are used for electronics, and now you can just put in different connections to make different chips, and now you can make a completely different device very quickly and easily. So that's kind of the concept here, that we can make a complex microfluidic device using a modular approach. So some of the manufacturing methods that are used for microfluidics, I'm going to just go through these quickly. We have photolithography, so for those of you that are familiar with that, casting is we take a material, you just pour it over the top of a mold, and then you can peel it off. And so that's really cheap and inexpensive, but you can't make things very fast that way because it takes a while for the material to harden up. Embossing, this is a material where you just press it into the device and leave an imprint. And then laminates, which is what I showed you just a moment ago, where you can cut out different shapes in a thin sheet and then push them all together, bind them all together. And then injection molding is very common if you have a large number of devices. So that's the best way to do it if you have millions of chips that you need to make, but it's very high upfront cost. And then finally, 3D printing I want to talk about for just a minute. So I think 3D printers are something that you may be able to get access to fairly easily. And if you have the access to the 3D printers, you're going to be able to do microfluidics much more easily than any of these other methods. So I'm just going to show you a couple of examples that we've done. So many of you probably have seen FDM printers. These are fused deposition modeling printers where they put in little drops of plastic. And one of the things you'll recognize very quickly when you're trying to make something with a 3D printer in microfluidics is the problem with the 3D printer is you're trying to make a negative space. You're trying to leave an opening, right? You're trying to make something that's not there. So that's actually hard. That's not what most 3D printers are built for. So if you take an FDM printer, you'll see, we'll start on this side. If you have a bigger channel, you're trying to make an opening through the space, the roof kind of sags in, as you might expect. So as you put these droplets of plastic over the top, it sags in and kind of deforms. And as you get smaller and smaller, this effect kind of gets bigger until it gets small enough that it doesn't really have a problem, but you can still end up with maybe different sizes than you expect. And that's kind of what this data is showing us here. We were targeting a 50 micron size. We actually told it to make 150, and then what happens is it adds the plastic in from the 3D printer, and it gets smaller. And we ended up with about a 40 micron size channel. So the nice part here is you can make very small microfluidic devices with an FDM printer. This is a cheap printer. It's like $500. But it has some limitations. So that's, but anyways, I'm hoping to be encouraging that if you can take one of these printers that you may even have here available to you, you can do microfluidics with it. So those are the cheapest printers, or FDM printers, but you can also use SLA printers, or the digital light processing printers. This is an example of a membrane, or a valve that uses a membrane to make in an SLA device. So this is, you have some pressure that's coming in, there's a membrane that's 3D printed, and then that 3D printed membrane is going to go up and down to block or allow flow through these channels underneath it. And this is what that looks like when you actually print it. These are ones that are 20 pixels wide, so at about 152 microns. And then you can make even smaller ones, in this case 6 pixels wide. This is a side view, and this is a top view to kind of give you an idea. And these are about 50 microns across. And again, this particular printer is a pretty nice one. It's very expensive, but there are printers that are much less expensive that can do similar things to this now. And this is how this device works. This is, I think I showed this a little bit earlier, where we have a valve. These valves are going up and down, and you can actually use it to pump fluid. So if you do these in sequence, now you're moving fluids around, and this can be 3D printed. So this is something that I think many of you would have access to that is not available in other, or the other manufacturing methods might not be available. You can take this same pump now and operate a whole bunch of pumps at the same time. So this is showing we have I'm just going to call it a dark fluid, mixing it with a light fluid, and then you get a gradient of these fluids across this channel. So you go from the dark fluids to the light fluids, and you can do chemistry tests with these gradients. So that's the example of a serial diluter in this case. Now, the other thing that 3D printing allows is some complexity. So look at this device here. In this case, we've made hundreds of valves on one array. This is a 50 by 32 array of valves, or 1,600 valves. And each valve is about the total area is about 20 millimeters by 12. And these are so small you can't see them. So if you zoom in on it, so if you look at this small space here, we have these different valves at each location. And so 1,600 valves were printed in this. So now you can have immense complexity with this device. And if you take it a step further, in this case, stack these up in the third dimension, you can build a device that has 11,000 valves in it. And that's this device here. This is looking at it from the side. You can build, just with a 3D printer, now we have a super complicated microfluidic device. Now, to lay this out by hand would take a long time. So I'm going to talk about maybe how to resolve that problem. But this device can be printed in 35 minutes. It essentially is perfect. There's no defects in it, and it can, in this case, we replicated it 5 times. This was a group, again, at BYU. And then the smallest microfluidic channel that was printed using this same printer is 2 microns by about 2 microns. It's not perfectly square, right? It's not exactly the shape that you want, but it can be printed. So again, maybe the take-home message here is we have a microfluidic device that can be 3D printed, and this 3D printer is probably something that may be a little easier for you to get a hold of than some of these other methods. All right, so thinking about complexity or simplicity, which of these would you rather have? A really complex chip or a really simple chip, right? So this is actually, well, the answer is probably both, right? Sometimes you need complexity, sometimes you need something that's very simple. And in this case, again, these are some kind of AI renditions of a complex chip versus a simple chip. But we're going to talk for a minute about how do we deal with complexity and maybe how can software help us with that. So if you look at these microfluidic, or we are working on a technique that we call microfluidic design automation. So how can we use software to help us design microfluidic devices? So I'll just walk you through this for a moment. So if you have a doctor comes to you and says, hey, I would like to do this test, they might give you this protocol of how this test is run. We're going to turn that into some sort of a language that the computer can understand. So, and then the computer is going to take that language and lay out a chip. So it's going to make pumps and valves and channels and heaters and other things like that and put them onto a chip. We want to make sure that this is going to work, so then we're going to simulate that chip. So we have a software that turns that into a simulation to verify that it works. You're then, once you kind of have it laid out, you can actually physically make it on a chip. We send it to a 3D printer, and then we can print that chip out and test it in a device. So this is some work that we're doing now. We're seeing if we can take these or take an assay and then print it essentially in just a few hours. So the idea, again, an idea for a chip to 3D printing it in just a couple of hours. So for those of you that do software, this is kind of our approach, and I realize you probably can't read that, so we'll look at it a little bit closer, but this is kind of our software approach to designing microfluidics. So in this case, we're going to take some software inputs. We need some sort of netlist definition, which is, here's kind of a list of the different components that we have in our microfluidic device. We have a component library, which is like all the different components that you could use. And this netlist is going to call some of these library components. And then we have some configuration for the simulation. This gets pulled into our software, and the software now lays out the entire device, simulates it, and then generates a CAD file that can then be 3D printed. So you'll note that, I may be a microfluidics expert, but I have to have software people to help me to do this. So if you're interested, I'd love to talk to more of you about maybe ways we can work together to use software to help us build these microfluidic devices. So we generate our CAD file. It goes to a 3D slicer, 3D printer slicer. We print it out. We have some sort of system that actually cleans out these chips now. That's called the auto flushing. And then we end up with our microfluidic chip. So it's now possible to have a computer design your microfluidic chip for you if you can tell it, this is what all I need to do is tell it what I want it to do, and it will figure out what components are required. So this is maybe a little more visual representation of the same thing. These are our inputs. We write some code. We have some CAD files we've developed in advance that represent the different structures. These are sent into the software. It puts all of these things together and turns it into a bigger CAD file that has all of the components in it that we can then print, and now we end up with that chip that I was just mentioning. So there's also some opportunities if we go back to this original kind of layout in what we're going to call the front end or the user interface. This is a place where maybe AI or some other things like that might be able to take that bioassay protocol and turn that into a language that the computer can read. So most protocols say mix this with that, heat it for so long, let it incubate for a while, and then mix this other. Anyways, it goes through the steps like that, and we need a way to take that natural language and turn that into something that a computer can use. Anyways, that may be something that those of you that work in software might appreciate. So we'd like to generate these designs from a standard input. So we've built some of these devices. So if you have a standard, you can go to a lab, and they have standard spec sheets on how to run their assay. And this is an example of that. It's not necessarily meant to be read, but it says mix this with that and do it in these ratios. So we collect the number of chemicals that we may need to use. We put that into a Python script. We write the software for that, and it generates a bunch of configuration files that then allow us to actually go through this process. So I think this the software design is something that really is going to be powerful in the future. So here's just one example of how this works. So in this case, we want to do we want to measure calcium in a urine sample. So what we need in that case is we need a sample. We have a reagent. In this case, it's called arsenazo, and then we mix that with water. And these are the concentration ratios that we need to be in. And that ratio is actually quite high. You'll notice that this number is much smaller than this one. So it's like a 1 to 225 ratio. We give this information to the software. It lays out all of the channels and other components, and we can then generate a chip that we can then print. And here's that chip after it's been printed. So we print the chip, we put it into this kind of a housing or a container to run the chip with all of we call it an interface chip that does the inputs and outputs from this. And now we should be able to measure calcium in urine. So we did that. Here's an example of how we did that with a couple of chips. And without going into this too far, we do get some sensitivity. It's a little bit less than if we do it by hand, but we can measure calcium, and this was on a chip that was automatically designed by the computer. So it works, is maybe the way to say that. So just maybe to kind of summarize a little bit of what we'd like to do with this, some of you may have seen 3D printing websites where you can go pick whatever you want to print. This is the Thingiverse website. I don't know if you've seen this one, but you can download all kinds of different things from Thingiverse and print them out. And so we would like to do something similar to that for microfluidics. So in this case, that would increase the accessibility by increasing the number of components. So instead of downloading a little fidget device or a little sculpture, you could download a microfluidic chip and print it out regardless where it was made. So maybe you design it, and I'm like, oh, I could use that. I'd download it and I could print it in the United States, or vice versa. I can design it and you would be able to download it and print it here. We've done this currently. We're working to integrate 8 urinalysis assays all together to work at the same time using these sorts of concepts. And so we'd expect that you'd be able to do that with even more of these devices. We have and then there's some optimization that can be done with this. But one of the things we need to do is we need to build these component libraries and modules, and that just takes a lot of people and a lot of time. And so that's one of the things where maybe you could join us. Maybe you could help us out. We could work together to identify what types of components we need, what types of libraries, and we can do this all remotely wherever we happen to be. So I'm going to jump to the end here for a minute and talk about just a couple things that I want you to know. So this first part is very important. So microfluidics can solve every problem. Do you believe that? Yes? Okay. If you do, you're crazy. It's a little bit of a joke, right? But microfluidics can be useful, right? So hopefully we can use it to solve a problem. As I mentioned, microfluidics is a tool. I use it that way, but it needs some help to achieve broader commercialization still. I'm very interested in, if any of you happen to be interested in working on these sorts of things remotely with us, I'd be happy to have that conversation. My email is there. You're welcome to reach out to me and we can talk about it. And so just want you to know that we need you. We'd love to work with you and figure that out. So I showed you some results today. I just want to show you, this is my research group back. Nice. This was our Christmas party in December. A couple of spouses there, but these are the students that do the work in my lab. And then these are some of the specific students that were doing this work. And then these projects were funded by these different groups. And maybe just in conclusion, I'd like to tell you 1 minute and tell you about the University of Utah. If you're interested in graduate school or other things like that, we have a wide variety of research programs at the University of Utah. This is specifically the Department of Mechanical Engineering, of which I'm chair. So we have programs in robotics and composite materials and things like that. This will give you an idea of the size of our department. But if you're interested in potentially coming to do a master's or PhD, please let me know. And maybe a couple pictures. I'm very grateful to be invited to come to India. This is what the weather looks like at my house right now. It's snowing. So, but this is my son and I when we went skiing last year. And we have some great sites and other things that you can come visit. And then finally, this is the University of Utah campus right here. So it's a little bit different than Chennai. And my lab is in this building right here. So I'm happy to answer any questions that you may have, and thank you for your attention.
We sincerely thank Professor Bruce Gale for the enlightening session on microfluidics, past, present, and future. Your talk provided a clear understanding on how microfluid technology has evolved and its increasing impact in biomedical and engineering applications. We truly appreciate your time, expertise, and the knowledge you have shared with us today. To express our gratitude, I now invite Dr. L. Sujata, Head, Center of Excellence in MEMS and Microfluidics, to present the memento to Bruce Gale.
You can keep it along with your